Hi, I am Ronak and I am from BioAero. And today we'll be discussing on pharmacogenomics. Now, what is pharmacogenomics? It's a combination of pharmaco plus genomics. Now, if we look at the term, it is self-explanatory that it is a combination of pharmacology and genomics. We all know what pharmacology of a drug is and what are the genomics. Now, if we pay close attention to functionality of pharmacogenomics, it becomes apparent that there is some kind of correlation between drugs and genes. However, there is more to it when we dive deeper into the subject. So there are two main terms when it comes to associating pharmacology with the genes. One is pharmacogenomics and the other is pharmacogenetics. While pharmacogenetics focuses on single gene drug interactions, taking into account allele genes, dominance and gene polymorphism in order to understand the better use of a drug on single patient or population. We have more information while we study the whole genome. It brings more clarity with the genome-wide association approach and epigenetics to deal with the effects of multi-genes or even chromosomes on the drug response. We are aware of the pharmacokinetics and pharmacodynamics, that is ADME, and what drug does to the body. Now, when we talk about ADME, it's absorption, distribution, metabolism, and excretion. When we take metabolism into account, we find that variations in DNA are responsible for alteration of its activity and the functional pathway. Perhaps an increase or decrease in enzyme and the transporters can alter the amount of drug present in the blood. We call this bioavailability. Let's understand this by therapeutic window. We have minimum safe concentration and we have minimum effective concentration. Based on the genetic makeup, the bioavailability of a drug can differ from individual to individual. The best example our industry uses to explain this is warfarin. Warfarin, an anticoagulant medication, is employed in the prevention and treatment of thromboembolic diseases. One of the challenges associated with this drug stems from its narrow therapeutic range, the propensity to cause bleeding, and the variable response observed in the individual patients when dosing. The attainment of safe and effective warfarin dose is a pressing and significant concern for numerous healthcare providers. Recent investigations have focused on variations in SNPs, also known as single nucleotide polymorphisms, within genes encoding two crucial proteins. One is the cytochrome P450-2C9 enzyme, and the other one is VCORC1, also known as vitamin K epoxide reductase complex. Now, CYP2C9 influences the metabolism of warfarin, while VCORC1 plays a role in pharmacodynamic response related to the expression of the enzyme target affected by the warfarin. Now, patients carrying the CYP2C9-2 and CYP2C9-3 alleles typically requires lower warfarin maintenance dose due to their reduced metabolic rate compared to those carrying the wild-type alleles. Similarly, patients with VCORC1 A haplotype typically need lower warfarin maintenance dose because of the reduced messenger RNA expression, which in turn leads to decreased production of protein required for VCORC1 formation. So this shows how the genes impact the therapeutic response in a human body. This is why it is really important for us to understand human genome that is present in our body. To better understand the responses that we are going to have, it's always better to have ourselves tested with whole genome sequencing, and then we can incorporate pharmacogenomics based on your DNA material, and it will be really helpful to lead a healthier life.